Hello and welcome to today's episode of Talking to a Biologist. Today we're with Dr. Dana Shroeder of the Department of Biological Sciences, and today we're in a teaching lab in the Boulder Laboratories. Dana, tell us, when did you first become interested in the biological sciences?
I suppose I always have been. I enjoyed being outside as a child and stuff, although my primary training is actually in biochemistry and genetics.
Biochemistry and genetics. So where did you do your undergraduate?
At Simon Fraser in Vancouver.
You moved from Vancouver to Winnipeg?
Actually, via San Diego.
You went from Vancouver to San Diego, and now you're in Winnipeg. Just to let everybody know, while we're taping, it's minus 40.
Exactly.
So what was your, where did you do your graduate work?
Calgary.
Calgary? And what was your PhD?
Actually, on worm genetics, C. elegans. It's another model system.
Model system. What do we mean by a model system when we talk biology?
Well, when we talk biology, we can't, well, eventually someday we'll understand all plants and animals, but often it helps if a lot of scientists focus on one plant or one animal and apply a lot of resources and know-how. And the idea being that if we can understand one plant or animal really well, that'll help us give us the tools to eventually understand a broader range of animals. So, for example, in animal genetics, mice, Drosophila, the worm C. elegans are good model systems there. And I'm currently working on a model plant, Arabidopsis.
Arabidopsis. And what does that plant look like?
Oh, why, look, here's some now. Actually, they end up much bigger than this, but the adults are about this high. But we can grow the seedlings on just a petri dish, which allows us to examine hundreds or thousands of them at a time, because numbers is an important feature of a good genetic system.
What do you mean by numbers are an important feature?
Often in genetics, we're looking for rare events. So if your population times are very slow or you have small populations, you're never going to identify that rare event. But if you have something that's easy and cheap to grow and you can grow in a small space, it's easier to look at hundreds, thousands of individuals to identify those rare events, which then will tell you about how the plant or animal works.
And that's why we use fruit flies instead of elephants, right?
Exactly.
Well, tell us what you've drawn here on the board. Is this part of that model system?
Yeah, so basically we're interested in how plants respond to their environment, because as we see outside in the minus 20 today, plants can't come inside like we did. They're stuck out there. They have to somehow adapt their physiology to temperature, light, a variety of environmental cues. In particular, we're interested in how plants respond to sunlight, both the visible light, which provides the food for the plants and is good and they like, and UV light, which is damaging to DNA and other features. And we're working on several proteins in the plant which are involved in both the good visible light reception by the plant and defending it from the bad UV light. And how we do that is as follows. When I showed that we can grow small plants on the petri dish here. Plants grown in the dark are tall and thin, whereas plants grown in the light are short and open their leaves and soak up the sun, like you would on a sunny day. We have a mutant, a defective plant, that even when grown in the dark is short and has open leaves, so it's soaking up the light that isn't there. So they're a little bit delusional, but we like that about them. So these tall guys are called etiolated, so these guys are called de-etiolated mutants, or DEET1 for short. And in my postdoctoral work, I show that DEET1 interacts with another protein called DDB1, which stands for damaged DNA binding protein 1, which, as you might imagine, binds to damage, in particular, UV damaged DNA. It does this with another protein, DDB2, and is part of the mechanism which ultimately results in repair of UV damage to DNA. Now, in humans, it's been shown that if this is defective, in particular if DDB2 isn't present, you get a disease called xeroderma pigmentosa, where you're sensitive to sunlight and have skin cancer conditions as well. So we're interested in what these genes are doing in plants and how they help plants repair UV damaged DNA, and also the interaction between this DDB2-DDB1 complex, which helps repair UV damage, and this DEET1-DDB1 complex, which helps plants respond appropriately to visible light, and the interactions between them that ultimately result in happy plants.
So by doing this sort of research, we can have healthy plants, have happy plants, understand how that works, and we can also apply that to human biology and things like skin cancer.
Exactly. And as well, for example, the tomato DEET1 and DDB mutants are called high pigment because they have higher levels of pigments like lycopenes that are actually nutritionally important. So yeah, this can be applied from everything to medicine to agriculture.
All right. Well, thank you very much, Dr. Shroeder. And that concludes today's episode of Talking to a Biologist. And your lecture will begin now.